Hello everyone, I'm back. We are starting our new unit on Mendelian genetics. This is a fun unit. It correlates with chapters 14 and 15 in your book. So please make sure you go ahead and at least this week read chapter 14 and maybe make it your goal next week to read chapter 15. This is going to be fun for you all because last year you would ask a lot of questions about genetics that I said we just can't go there yet. And this year we get to go there a little bit. That's the nature of AP Bio. We get to look at things in a little more detailed way and answer some questions that you just weren't ready for. And I think you heard enough last year that genetics is not as simple as brown hair is dominant over blonde hair. It gets much more complicated than that. And that's what we're going to look into. But to start, let's start with the simple. Let's start with the Mendelian genetics. Mendelian genetics is the idea that one trait is dominant over another. And so last year we talked about it, the pea plants. This idea that if you had, and remember he used pea plants because they were able to self-fertilize and it was quite wonderful. But if you had yellow peas and you had green peas, and the yellow peas would be the dominant, and this first generation, and you do need to know this now for your test, just to be able to speak the language of genetics, this idea that you have three generations a lot of times in genetic studies. You have the parent generation, and it's indicated by the P. You have the first generation, and it is indicated by F1. And then you have the second generation, which is an indication of F2. And that's, oh, I've got a cat that'd like to make a cameo, but we're going to tell him no way. So F1 is the first generation, F2 is the second generation, and the original breeding was is known as the P generation. In this case, you see the yellow peas were dominant because the second generation, all you got were yellow peas. But hiding underneath are the green alleles that suddenly pop out. And then on the second generation, we have a 3-to-1, 3 yellows to 1 green phenotypic ratio. So this demo, I'm hoping that it shows up as this cat, oh, sorry, buddy, on my screen. Looks like the cat kind of messed that up, but I'm gonna rewrite it. So this is just this little bee demo, and it's if we have R's, let's make R the dominant color. So this would be big R, if you guys see the red beads. And then the yellow beads would be the recessive, so little r. So what would the P generation be in this mating? It would be big R cross with little r. So just in case I never know if things are going to come through, it would look like this, big R times little r. And this would be the red, and this would be the yellow, okay? And this is how you would write the cross. What would be the color of the F1 generation? Well, red is dominant, right? So the F1 generation would look a lot like the big R, except the genotype would be different. So the F1 generation, when we cross the red and the yellow beads together, would look like this, big R, little r. And just to show you, their offspring would look like this, because this was what their offspring would look like, because all that the big guy, that the whoever this is, this one could give would be a big R. And all that this one could give would be a little r. So we end up resulting in big R, little r as all the offspring. However, their color would still be red. Everyone see that? All right. What would the color of the second generation? Well, the second generation, we're going to have a whole bunch of different things going on, aren't we? The color would be some red and some yellow. So it looks something like this. Red and yellow beads mixed together. Finally, out pops the yellow allele. And all the different genotypes, we would get a couple different ones. Remember, you've got to draw your Punnett squares for this, but the second cross would be represented by this F1, big R, little r times big R, little r. And what would you get out? Well, if you go and you quickly, this is really just meant as a review, you quickly draw your Punnett square, you end up with the following genotypes. You have 2 heterozygote reds, big R, little r's, and you have 1 homozygote dominant red and 1 yellow, okay? So where does the recessive trait go in that F1 generation? Well, it was all red, right? So it looks like this in that F1 generation. However, where did the yellow guys go? Well, they were hiding. They were hiding behind that dominant R. Okay, the little r was just hiding underneath. All right. So here's another quick demo. I don't have it for you, believe it or not. It's in the afternoon and I don't have any coffee. But if it were the morning, I would certainly have that coffee. But this is an example where, okay, if you have coffee, you could have black coffee or you could have like mine, where you have a lot of cream. Now the cream dilutes it a little. Now this question is, does this really exist? Could we make this as a comparison with genetics? Is it always one or the other with genetics? Well, you guys already know that's not the case. Let's think about things like skin tone and hair color. It's not just like you're black or you're white. There's many tones. So yes, you have something, this is what we're going to get into later in this unit, where you can have something that's in between. And we'll talk about that as we move on in the unit. All right. Oh. I didn't realize I had that cool sound effect, but that is very fun. But I'm just going to turn it down a little. So alleles are different forms, this again is review, different forms of an alternative gene. How do alleles, and obviously you just need to know this stuff at this point, how do alleles relate to homologous chromosomes? Well, if you have homologous chromosomes, will have the same genes, but they might have different alleles unless, you know, you've got your identical twin as who you made it as your mom and your dad, but it doesn't work that way because you have to have different sexes. So it just doesn't work. But alleles are different forms, or they could end up having the same allele for a gene. So here, this is flower color, right? So these are homologous chromosomes, and you're always going to get 2 copies of each gene. And the allele, that's a different way we could say it, you could have 2 different alleles. This one, this little flower, has an allele for the purple flower color, and they have an allele for the white colors. So we could say that homologous chromosomes may contain different alleles for the same character trait. Right here, and this is the key thing, homologous chromosomes may contain different alleles for the same character trait. All right. Oh, here's a fun table talk. If in the canine population there is a whisker length gene, there are only 2 possible variants of this gene: an allele for long whiskers and an allele for short whiskers. Now, there's a mistake here. This should say, how many whisker length alleles does an individual have in one of her somatic cells? So obviously this is right off the test because look, I left eyelash length instead. So in the canine population, there's a whisker length. you can only have two possible variants. You can only have a big L, and that equals long whiskers, or a little L, which would equal short whiskers, okay? So if the individual, how many alleles does the individual have? Pause it if you want to think about it and answer it yourself. But I'm going to go ahead and answer that for you right now. Here's a chromosome. Here's the allele for a whisker length. These are homologous. I'm drawing it. I will show you. And this represents one allele, and this represents the other allele. All you can have for a pair is two alleles, okay? Because they say there's only two possible variants. This is a question that gets a lot of people on the test, okay? An organism with two of the same alleles for a particular trait, we call them homozygous. An organism with two different alleles for a particular trait, we call heterozygote. Okay? That should be review again. These are easy peasy things. Remember, the dominant allele masks the effects of the other allele, and it's usually we select the dominant allele as being the uppercase letter. The recessive allele is hidden if the dominant allele is present. And it's always designated by the same letter as the dominant allele, but it's now a lowercase. So I don't think you can see here on this cat, but short hair is actually dominant to long hair. So the allele, short hair, would be the capital S. I like to also do for something like S. I make the dominant one the uppercase capital and the recessive the lowercase of S, but cursive, so that you can really tell the difference. But short hair could have two different genotypes. It could have big S, little s, or big S, if you see. This would always equal the short hair. Now, long hair is a different deal. That's the recessive allele. So in order to show it as the phenotype, it must have two little s's. So this beautiful, I love long-haired cats, must have two little s's for her genotype. Okay? Again, phenotype